Hi there. Welcome to the variant calling course using the Linux operating system. In this course, we will be dealing with the bioinformatics pipeline for analyzing the genomics data, where we will get the reads file from SRA form or FASTQ form to into variants file. So basically what all this heading was, what variant calling is, what are variants, how to deal with variants, what is bioinformatics pipeline, and also basically what all this course is mainly about. So dividing this course, if you're familiar with the NGS data, you know that the next generation sequencing data produce a lot of files, and by using that files we can analyze the genomic variants, like how were the changes that were defined in the previous era are now defined in the reference genome. For example, if our reference genome contains the site of thiamine that is changed in our reference file to the adenine, then variant calling is called variant calling. That how we call the variants. So for calling the variants from the NGS data, we need a specific Linux pipelines. So we have to work with Linux operating system as well. So if you are using Windows, I'm gonna make a Windows subsystem as well. Gonna include a whole section of command line in bioinformatics as well for you to get ready your Linux operating systems as well. So we're gonna retrieve some NGS files. After retrieving NGS files, we're going to retrieve some variants from it. By retrieving that kind of variants, we are going to analyze that data. We are also going to check the quality of our data. We are also going to trim that quality to get our maximum good quality data from the NGS files. So discussing the table of contents for our course, our contents are mainly on the NGS based data. For example, we are going to deal with what basically variant calling is. I'm going to include a lecture of that as well. So what is background and metadata we are using, understanding the data sets and hypermutability and accessing the read quality. For example, checking the quality fast QC, trimming that quality, applying the fast QC on your data as well, applying the port job on your data as well. After getting that kind of data, we are going to involve in some steps for variant calling pipelines. We are going to develop a pipeline as well. I have already developed the code for you guys, so you just have to copy all the code steps and run onto your Linux terminal. So the tools that we are going to use, we are also going to discuss that as well, so that is also included. So that was it for the short introduction of our course, what basically variant calling is, how to call variants, and what basically the title of this course is. So if you're interested in learning about variant calling, if you're interested about getting to know about your reads, how to extract features from your, how to extract variants from your files, variants from your NGS files. So this course is for you, and this course will help you to understand. I hope that this would be enough for my introduction video. That is it for this lesson, I think.